Hello, Dr. Williams here. I'm going to just go through some B5 homeostasis and response with you, hopefully recapping a lot of what you've learnt in order to help you prepare for an exam or a test or your paper 2 for your AQA combined science exam. This is the combined science content. There is actually a lot of triple only content, but we don't cover this on the video. What we will cover is everything you need to know for this unit, for your foundation and higher combined science. Let's get started. Okay, so the first thing that you're going to need to understand is the definition of homeostasis. Homeostasis is the regulation of the internal conditions of a cell or organism to maintain optimum conditions for function in response to internal and external changes. You have to know that definition, but the bits in bold are the most important. The fact that it refers to internal conditions, and that could be either of a cell or organism, to maintain the optimum conditions. Optimum means ideal. Only in these ideal conditions can our cells really function properly. So make sure you learn that definition because you could be asked just to recall it. Once you know the definition, you also need to have some understanding of the internal optimum conditions in the human body. So for temperature, that's 37 degrees. The pH is 7.2, and that's controlled by your water and iron content in the blood and all the tissue fluid. Glucose concentration is around 90 milligrams per 100 centimetre cubed of blood. You won't have to be able to recall that, but it's really useful to be able to have some sort of ballpark idea of how much glucose you need in your blood to function properly. All the mechanisms of controlling these things are actually automatic. So when we regulate things by homeostasis, like temperature and pH, they are automatic. And in these automatic control systems, you might use either the nervous system or the endocrine system. And the nervous system has our nervous responses. The endocrine system uses chemicals, often called hormones, which are used to communicate throughout all the different parts of the body. We'll look at both systems in turn as they come up. So what really is homeostasis and how does all of this work? No matter which system you use, the nervous system or the endocrine system, you're going to use this similar pathway. And the pathway starts, if you look at the diagram, with a stimulus. And a stimulus is just a change in our environment. For example, that could be a light coming on. Receptors then detect this change. So the receptors here, concentrated in the eye, they would detect the change. And that information is then transferred to a coordination center, which in this case would be the brain. But it could be it's the brain or the spinal cord or the pancreas, depending on which condition you're going to regulate. Now, the coordination center's job is to receive that information from the receptor and then process it in order to bring about the effect. So it then acts on effectors, which are muscles. In this case, it's the muscles around the pupil. But it can be a gland that also brings about an effect, especially when you're looking at the endocrine system. So the effector then will bring about the overall response. That's how your body reacts or responds to that stimulus, and that restores the optimum levels. So all homeostasis pathways follow this main pathway of events. It just really depends if you're looking at the nervous system or the endocrine system into what happens at each stage. If we think about our main ways of sensing the world around us, we have got sense organs, and these include many receptor cells which can detect all the different stimuli. Now, you'll be familiar with these from primary school, where you learnt about the sense of smell and the sense of sight. But your sense organs, which detect all of those things, are the eyes, ears, nose, tongue, and skin. Your eyes will contain light receptors detecting colour and light intensity. Ears will contain sound receptors and also your position, so it also coordinates your balance. The tongue and nose are actually receptors which detect chemicals. So although you have a sense of smell and taste, you're not detecting smells, you're detecting the chemical that your brain interprets as that particular smell. The skin is a very complex organ that detects touch, as well as more harder pressure, pain, and the temperature. So all of these sense organs are really vital for helping us to respond to our surroundings. So one of the main ways that we respond to our surroundings is with the nervous system. And the nervous system actually coordinates all of the information from what is happening in the world around us. And this information from those receptors will actually pass along cells called neurons, which are basically nerve cells, but their scientific name is neuron. And they pass through them as electrical impulses from the receptor to the central nervous system. And it's the brain and the spinal cord that form the central nervous system. The nerves which branch out from the spinal cord and the brain is called the peripheral nervous system. So you don't need to know that terminology specifically, but just know that it's all of the neurons that actually feed the information into the central nervous system. And neurons or nerve cells are very specialized cells, and they send electrical impulses from those receptors to the central nervous system, or CNS. And then they're also involved in sending the electrical impulse from the central nervous system to the effectors. And to do this, they're very long and thin, and they have many branches. And that means that they can carry electrical impulses over very long distances. They can grow up to 1 meter long. They also have many mitochondria because they need to aerobically respire in order to release energy and allow the electrical impulses to travel along them. And they actually have a fatty myelin sheath, which goes all the way around the central part of the cell. And the central part is called the axon. And that's where the electrical impulse travels. So it actually insulates the electrical impulse, and it allows the electrical impulse to travel faster through the neuron. And there are 3 main types of neuron, but they all use those mechanisms and have those special adaptations. You have a sensory neuron. Now, these are connecting the receptors to the central nervous system. So our sensory neuron connects the receptor to the central nervous system. A relay neuron finds in the central nervous system itself, and it sends electrical impulses from a sensory neuron to a motor neuron. Now, these are only involved if there's a reflex arc. You need to be able to name that neuron if you're looking at reflex arcs, which we'll look at in a minute. And then you have motor neurons, and motor neurons, best way to remember is help to get you motoring. So they send an electrical impulse to the effectors from the central nervous system. So really, the sensory neurons help you sense, that's from receptor to central nervous system, and the motors help you move, or motor, and that's from the central nervous system to the effector, which remember is normally a muscle or gland. The relay neuron only comes into play when you're looking at reflex arcs. And when we think about a reflex arc is actually something which comes under the unconscious response. So the way we can respond is by thinking about it or having a conscious response. So you coordinate it through the brain and you get the information from the stimulus and you decide what you're going to do about it. So, for example, if you see an apple and you feel hungry, your eyes, the light receptors will send the information to your brain that there's an apple there, and you will decide whether you're going to send an electrical impulse through your motor neurons to your muscles in your arm in order to pick that apple up and eat it. An unconscious response, though, or a reflex arc, is something that's automatic. So it's usually in response to something painful. So if an apple sort of hits you on the head, then you might try and have an unconscious response of a blink response where you don't need to think, oh, I must blink to stop this apple from hitting me in my face and hurting my eye. You will do that automatically. And that's an unconscious response or a reflex arc. So if we look at those unconscious responses in a bit more detail, they are reflex actions, and that means they don't involve the brain. These are usually in place because they are automatic and rapid, and they help us to stop us damaging or harming ourselves. And the reflex arc is basically a series of neurons which bring about a reflex action. So, for example, if you're going to touch a hot pan and you don't realize you touch the pan, and immediately receptors in your skin will detect that the pan is hot, and a reflex will happen where you move your hand quickly away. if we're looking at that response in a reflex arc, this is really what it looks like. If we just have a little look at this diagram, you can see that there are receptors here. So that's a very generalized receptor. So it could be a receptor in your skin, for example, if you're touching the hot pan. So when you get that stimulus of touching the hot pan, the receptor will detect it, and it will detect it through its pain receptors. Then the information from the receptor will be sent as an electrical impulse through the sensory neuron. So remember, that's the neuron that connects the receptor to the central nervous system. Now, because this is a reflex, this information isn't going to the brain, it's actually going straight to the spinal cord. So that's where your arm connects. So sort of probably at shoulder height, if it was your hand touching a pan, it will go straight into the spinal cord where there's a relay neuron which connects the sensory neuron to the motor neuron. So the electrical impulse has gone down the sensory neuron and now has been passed into the relay neuron. And that relay neuron then sends an electrical impulse through the motor neuron, which, remember, the motor gets you moving. So that will connect to a muscle in your arm. That muscle will contract and you will lift your arm away from the hot pan. You can see in this example that the spinal cord is situated in the central of your spine. So your spine is the bones and the spinal cord is a very dense region of all of these neurons. And you can see there's also some gaps here labelled as synapses. And these are little gaps that connect the different neurons, which we'll look at that in a minute. There is a note though, so it does say if the reflex is near the brain, for example, if it is the blink reflex rather than skin on your hand or something like that, the relay neuron may actually be inside the brain itself because it's the quickest path to where that reaction is happening. It doesn't have to then go into the spinal cord if it's only happening in your eye, for example, if your eyelids need to blink in an automatic response. Okay, so let's have a little closer look at these synapses. So the synapse, remember, is the gap between the neurons, and the gap between these neurons is just called the synapse. And the electrical impulses inside your nerve, so here's a sensory neuron, it's actually connecting to a motor neuron, but there is a synapse between them. The electrical impulse goes through the neuron, and at the synapse, basically we have chemicals that transfer the information from one neuron to the other through that synapse or gap. Then that stimulates another electrical impulse around the next neuron. So we can't have electrical impulses going across the gaps because obviously we've got a lot of liquid in our body, mainly water, which will then take the electrical impulse away. So the water will conduct the electrical impulse away. So we need a different mechanism at our synapses. So let's have a closer look at the actual synapse. So these end bits of the neuron are actually called dendrites. So this is the end of one of those branches of those neurons, just here on the diagram. And then this other one could be a dendrite of another neuron, or it can be actually the muscle or gland, the effector itself, where the synapse goes. So this is the pathway of what happens. You'll see the chemicals are first of all stored in these tiny vesicles that's in the cytoplasm of the neuron. And these chemicals are released when an electrical impulse passes through the neuron. The chemicals are released because they get fused with the membrane and they are released into the synapse or the gap where they will diffuse from a high to low concentration across the gap. As they get to the other side of the gap, they will bind to the receptors which are part of the next neuron or effector. And these receptors are specific shapes so that the chemical will fit inside the receptor. And once that's there, you will initiate another electrical impulse through the other neuron, or you initiate the muscle to contract or a gland to release its hormone if it's acting on an effector. So a synapse is really the gap where the chemicals diffuse across between the different neurons. Here's an exam question just to have a look at how this all links in together. So this is the doctor tests a reflex action of a patient. When the doctor touches the patient's foot with a blunt rod, the patient's toes curl. And it asks you to describe the pathway of this reflex action. So there is no thought involved in this. This is a reflex, so it's automatic. So this is the kind of things that you would need to say. The receptor detects the stimulus, in this case it's being touched with the blunt rod. The electrical impulse travels from the receptor to the central nervous system along a sensory neuron. Chemicals are released by the sensory neuron and they diffuse across the synapse. Then an electrical impulse travels along a relay neuron in the central nervous system. Chemicals are released by the relay neuron, which diffuse across the synapse. And this makes the electrical impulse travel from the central nervous system or the spinal cord to the muscle along the motor neuron. This goes to the effector, which makes the response of the toes curling, or to the muscles in the toes, which then contract. You can say either of those things. So that's kind of a summary of the level of detail that you need to know in order to describe a reflex action. There is a required practical in this topic though in B5, and it's really important that you're able to look at this and be able to plan an investigation in order to measure how quickly you react. Now, you're not going to plan an investigation to measure how your unconscious reaction times work, so in other words a reflex arc, because that would be dangerous. Remember, reflex arcs are there to prevent you from harm or injury. So we don't want to use any automatic response like that because it could cause you harm. So we use this to measure our conscious reaction times. And remember, our conscious reaction times is when information goes through the brain and we decide on what to do in order to respond to the stimulus. So they involve both sensory and motor neurons, but a lot of other neurons in the brain. Now, you can measure the reaction times. You can use a computer program, which would be more accurate. So, for example, pressing a button when you see a certain shape appear on a screen, or the ruler catch method, which is less accurate, but it is easier to carry out. If you're doing the ruler catch method, you basically need a partner. One person sits down on a chair and the other person gets a meter rule. The person holding the meter rule will hold it in front of the person sat down, and that person will look straight ahead until the ruler is randomly dropped. The person sat down then catches the ruler and records the distance the ruler is caught and uses a conversion table to calculate the reaction time. They then have a rest, and this needs to be repeated at least 10 times, and all the results must be recorded. Then you would change a factor that you wanted to investigate and repeat the whole thing again. So do 10 repeats of your reaction time. And the reason you've got to record all the values, even if they don't look right, is to avoid bias. And you can calculate the mean reaction time before and after changing the factor. These are all possible things which could be changed in this sort of investigation. The amount of caffeine you have, or sugar or alcohol, if you've exercised or you exercise regularly, how tired you are, your age, the practice performing a task or your fitness, your distractions, there's someone trying to talk to you, distract you. You might have excess lactic acid from anaerobic respiration. You might have a different temperature environment that you want to investigate. Lots of different variables which you could see if they affect your reaction time. But whatever you change is called the independent variable. And you're measuring in this investigation the distance caught on the ruler in order to find the reaction time. That then becomes your dependent variable. All the control variables are all of those other things that you need to keep the same. For example, the age of the person, the people that you're investigating, or if you're investigating the effects of caffeine, you need to give everybody a drink, but some with caffeine, some without caffeine, to make sure that actually it's not the liquid itself that's affecting the reaction times. So if you're doing caffeine consumed, there are lots of different control variables, not just in kind of the other things that you could do, for example, if you've eaten glucose, but also in the way you've carried out your investigation. So the height the ruler is dropped from, whether you use your right or left hand, or your dominant hand or your less dominant hand, if you rest your arm on the table or not, all of these factors need to be controlled. So it's a really good investigation to make sure you understand exactly which variables you're changing, measuring, and controlling. If you do this investigation, it's also a really good way to analyse it if you plot it on a bar graph. And if you plot it on a bar graph, you can plot the mean on the bar graph, but also the range of the lowest to the highest value. And then the range bars can be compared to see if there is a significant difference or not. The way you tell if there's a significant difference is if you look for an overlap. So for example, here on the right of this bar graph, you'll see there's an overlap in ranges. What that really means is that student B's before caffeine value could have been anywhere within that range, and then the after caffeine value also is anywhere in that range. So there's no difference between before and after caffeine. So you would say there's no significant difference in those two sets of data. However, this one, there is a difference. The range bars do not overlap. So for student A, the caffeine seemed to have a greater effect on their reaction times than the student B. If you do take note though, a shorter reaction time means a lower value, which means the reaction time is faster. A longer time is a bigger value, which means the reaction time is slower. So be very careful when you're looking at data. A shorter reaction time means the reaction time is faster. Avoid using terms like better and try and stick to faster or slower. So I said there were two different mechanisms of homeostasis. One was endocrine and the other was nervous system. We've looked at the nervous system and how we respond to the environment using that. The other system is the endocrine system. So that's our hormonal system. And so remember, homeostasis is regulating the internal conditions of a cell or organism. And sometimes we need to do that very quickly, which is where the nervous system comes in. Sometimes this could be more slowly, which is where your endocrine or hormonal system comes in. So if you look at the nervous system, we are going to be using electrical impulses sent through neurons and chemicals at the synapses. Whereas in the hormonal system, we have some glands which secrete the chemicals, and these chemicals are called hormones, and they're carried through the blood to target organs where they need to act, because the blood will flow everywhere around the body, so it will get to the target organs through that pathway. The brain and the spinal cord are the key parts of the nervous system. In the hormonal system, you have different glands which are involved, of the pituitary gland, adrenal gland, ovaries, testes, pancreas, and thyroid. Information is very quick through the nervous system. It enables us to respond very quickly to our surroundings, whereas our hormonal system tends to be slower. So we're talking 0.2 seconds for a coordinated response in the nervous system, whereas in a hormonal system, it could actually be years. For example, puberty lasts over a long, long period of time. The information as well, when your nervous system has acted, that's it. You contract your arm, for example, to move it away from a hot object. It doesn't stay contracted. You instantly can move it again. Whereas the hormonal system, longer lasting effects. So for example, again, in puberty, once you've gone through the stage of puberty, those effects keep lasting. And in the nervous system, examples that you've got there are reflex arcs, or the ruler reaction times test, or regulation of temperature. They all involve the nervous system. Whereas the hormonal system, we have hormones like testosterone and estrogen, adrenaline, insulin, and it's also how we regulate water and blood glucose. So we're just going to look at blood glucose in a little bit more detail. But before we do, we are going to have a little look at all of the organs involved in the endocrine system. So you need to be able to label these organs and say where they are in the human body. You'll see you've got a master organ called the pituitary gland right at the top near the brain, and that's called the master gland because it controls and coordinates all of the other glands in the body. The pancreas, just below the stomach, you'll be familiar with that from B2, where you learn about its role in digestion, but it also regulates blood glucose concentration. So it releases hormones called insulin and glucagon into the blood. Sorry, the pituitary, I should have said, also has hormones called FSH and LH, which are released from it. It releases many different hormones, to be honest, but these are the two that we're going to be looking at later. Our ovary, obviously only found in women, they release estrogen and progesterone hormones, which you've probably heard of before. But you also have a gland called the thyroid, which is just in the neck, and that releases a hormone called thyroxine. You would need to know about that if you're doing the higher tier paper. Adrenal glands, just on the kidneys here, will actually release adrenaline. Again, you would need to know about that function for the higher tier paper. And in males, we have testes, which are the male glands which release testosterone. Again, you've probably heard about that hormone before. So let's just have a look at some of those higher tier hormones that you need to learn about. So adrenaline is produced by the adrenal glands in times of fear or stress. And its role is to increase your heart rate and boost the delivery of oxygen and glucose to the muscles. That prepares the body for flight or fight. This allows more aerobic respiration in muscle cells, so there can be more muscle contraction, and you can run away from something that might be frightening you very effectively, or defend yourself if you need to fight. The other higher tier hormone is thyroxine, and in a similar way, it does affect reactions, but it actually affects all of the reactions inside your cells. So it will include respiration in this, but all of those other reactions. And all the reactions inside a cell is called your metabolic rate. So it actually stimulates the base or basal metabolic rate. And this is really important in order to help humans grow and develop. So that's the role of thyroxine from the thyroid gland. Again, that is just higher tier. However, for higher and foundation tier, you need to know a little bit more information. Now, if we just slow down and have a look at the slide, I know there's a lot of information on this slide, but this is really how hormones regulate your blood glucose concentration. So never call it blood sugar, it's always blood glucose. So let's stick to glucose. So if the optimum is shown by this arrow going through the middle, that would be 90 milligrams per 100 centimeters cubed of blood. If you've eaten a large meal or had a sugary drink, your blood glucose concentration may increase and it will go higher. That is actually detected by receptors because that's a change in the stimuli, isn't it? It's change in the stimulus. So receptors in the pancreas will detect this increase in blood glucose. And it's actually cells in the pancreas that then coordinate the information and release a hormone called insulin. So no involvement of the brain here. It's the pancreas that's the coordinator. And it releases this hormone insulin. And insulin goes into the blood and it travels all over the body. But when it gets to a target cell, which is either liver or muscle cells, it binds to receptors on the target cells. And the glucose, this stimulates those cells to take the glucose in from the blood to the cells, where they are converted into a storage molecule called glycogen. So you came across glycogen in B4, where you heard it was a storage molecule, and so it's always a store of glucose when your body needs it. Because the glucose has been taken out of the cells and stored in the liver and muscle cells as glycogen, the blood glucose concentration has now lowered. So your response has been to lower the blood glucose concentration and you're back to the normal levels. Now you need to be able to describe that pathway if you're doing the foundation paper. However, if you're doing the higher paper, you need to also be able to describe the opposite response. So when you've responded to high blood glucose concentration and you've lowered the blood glucose, you're actually in a condition where you've now got low blood glucose concentration. So again, this is detected by receptors in the pancreas. They would detect the decrease in the blood glucose. The cells in the pancreas coordinate the information and release a hormone called glucagon. So a different hormone altogether called glucagon. This glucagon binds, again, goes through all the blood, all around the body, but it binds to receptors on target cells, which are liver and muscle cells again. And this makes the glycogen convert into glucose. And the glucose is then released back into the blood, and that will increase the blood glucose concentration. So what you have to know for high blood glucose, receptors in the pancreas, cells in the pancreas coordinate the information and release insulin. Insulin binds to target cells, glucose is taken in and converted to glycogen, bringing it back to the norm. Low blood glucose concentration, receptors in the pancreas coordinates information and releases glucagon. Glucagon binds to the target cells on liver and muscle, so glycogen is converted to glucose and released back into the blood. This raises the blood glucose levels back to the optimum. In actual fact, what your body does is when you eat things, it goes through this and then it goes straight through the other way because it goes beyond the norm. So it's constantly releasing insulin and glucagon in order to maintain optimum blood glucose. There are some really key terms then for you to get your heads around for this. You have to know glucose. So I said before, never talk about blood sugar. It's always glucose. Glycogen is the insoluble storage molecule which you make from the glucose monomers inside of cells, and that stores the glucose away. The pancreas is the gland which has the receptors which detect the blood glucose concentration, but it also coordinates the release of insulin when glucose is high or glucagon when glucose is low. So insulin is the hormone released when the glucose is high. Glucagon is a hormone released when the blood is low. So you could think that the glucose has gone to help you remember glucagon. And the reason we have to do this, because glucose has quite a horrible effect on your cells and osmosis. So that's the movement of water in and out of cells. So glucose needs to be transported for your cells to use in aerobic respiration, and it's small and soluble, so it can move easily in and out of cells, but you can't store it in cells. If you tried to store it in cells, it would affect osmosis. Because it is soluble and it increases the concentration of solutes in solutions. So when you increase concentration of solutes like glucose, it means water will move from the dilute to concentrated solution by osmosis, and this will cause cell lysis. So you can't have high glucose concentration in your blood because water will move out of your body cells into the blood and they will actually be destroyed. So you must regulate the glucose concentration to make sure osmosis isn't occurring and damaging cells. And that's why we need to store glucose as glycogen. So if there's excess blood glucose, the liver and muscle cells can store its glycogen. Glycogen is large and insoluble. It doesn't move through cell membranes. It can be stored. And because it is insoluble, it doesn't affect osmosis. And glycogen is easily broken down. It's a large, complex molecule, and it can be easily broken down into its small glucose monomers, we'd call it. This will happen in liver and muscle cells. If the glucose leaves the cells and enters the blood, it can rapidly increase blood glucose concentration. So it's because it's such a large molecule with so many glucose monomers on it, you can rapidly increase glucose concentration as you need to. However, sometimes this doesn't quite all work the way we want it to. So that can lead to disorders like type 1 and type 2 diabetes. So type 1 diabetes is a disorder where the pancreas doesn't produce enough insulin. There's something gone wrong with the pancreas. It doesn't produce enough insulin. So you'll have high blood glucose concentrations, which can only be brought down really by being injected with insulin. So you have to have regular insulin injections in order to control your high blood glucose concentration. Low blood glucose concentration, if you're diabetic, is easy to control because you can just consume more glucose in the foods that you eat. It's when it's high, we can't easily remove the glucose from the blood. So in type 1 diabetes, it's much more likely to develop in adolescence. That means sort of as a teenager. And there is some suggestion it could be due to an autoimmune response. So that's basically your immune system cells actually attacking the pancreatic cells and stopping them from releasing insulin. But it can be managed with insulin injections and a healthy diet and exercise. Type 2 diabetes, on the other hand, is different. So you can see this pathway here. It's not the pancreas being unable to make insulin. It's the fact that your cells don't respond to that insulin properly. So it's a disorder where the body cells no longer respond to insulin, even though insulin is being made by the pancreas. So again, the symptoms of it would be high blood glucose levels, and you can normally control type 2 diabetes by controlling the carbohydrate intake that you have. So more complex carbohydrates are better for you because they digest much more slowly. Any sugars in like sweet drinks and things like that, that would be difficult to manage if you're type 2 diabetic. Excuse me. So in type 2 diabetes, some risk factors for it are obesity. It's much more likely to develop in older adults, and it actually can be more regularly controlled by diet and exercise. Okay, so that's all of the how we regulate glucose and the involvement of the pancreas as an endocrine gland. We're now going to look at some of the other endocrine glands by looking at the reproductive hormones. So your secondary sex characteristics, that's basically the puberty characteristics, are controlled by different hormones in males and females. In females, it's estrogen, which is the main female reproductive hormone, and that's produced in the ovary, and that causes your secondary sex characteristics to develop into a female. So you will get eggs beginning to mature in the ovary. One egg is released every 28 days when you reach puberty, and that's called ovulation, and it's all down to estrogen being released causing those effects to happen. In males, when they go through puberty, is testosterone, which is the main male reproductive hormone, and that's produced by the testes, and that stimulates sperm production as their main feature. In females, when they go through puberty, they often start the menstrual cycle, and the menstrual cycle is a series of events that actually helps you to be reproductively viable as a female. So the menstrual cycle lasts about 28 days, and it starts to happen when women reach fertility. The cycle starts on day 1 as the uterus lining breaks down. That's often called having a period, and this uterus lining will leave through the vagina. There's a diagram over here of the female reproductive system if you're forgetting some of these parts. Excuse me. So there's the ovary, which is the glands there, the oviduct, which is a tube which the egg would normally travel down to become fertilized. The uterus is where a baby would develop, or a fetus would develop inside, and that would grow inside. The cervix is a strong muscle which holds the fetus in place as it develops, and the vagina is just basically the channel that the baby comes out when it's born, and also where the uterus lining will leave when you're in the menstrual cycle. Menstruation, when the uterus lining starts to break down, often lasts around 5 days. During the menstrual cycle, if it starts on day 1 when the uterus lining breaks down, after about 5 days, the uterus lining will start to build up again. And at day 14, one of the eggs is released from the ovary, and that is called ovulation. And then if that egg is not fertilized, the menstrual cycle will restart again after around 28 days when you get the menstruation happening and the uterus lining breaks down again because there isn't a fetus in there to develop. All of this is controlled by a number of different hormones. You have the pituitary hormones, which are called FSH and LH, as well as ovary hormones. So we'll look at the ovary ones in a minute. We're just going to look at the pituitary hormones. So even though the pituitary gland is up in the brain, the hormones FSH and LH actually act on the uterus lining and the ovaries themselves. So FSH job is just to cause eggs, which are in the ovary, to become mature. It also, if you're doing the higher tier, you have to know that it stimulates the ovary at the same time to produce estrogen hormone as well. LH will cause eggs to be released from the ovary. And if you're doing the higher tier, you need to know it stimulates the ovary to produce progesterone, that other hormone. So FSH causes eggs to mature, LH causes eggs to be released, and then the interactions with other hormones are what you need to know for higher tier paper. So let's have a look at those other hormones, the ones made by the ovary. Here you can see we have estrogen, which builds and maintains the uterus lining. It also stimulates the pituitary gland to produce LH, which remember is that hormone that makes eggs be released. And it inhibits or stops FSH production. Now that sort of makes sense really. If you're going to build and maintain the uterus lining, you're getting ready for a pregnancy. What you don't want to do is produce further eggs. So to stimulate, produce LH will actually, it might make eggs mature, but it will stop them being released, okay? You've also got progesterone released by the ovaries, and they help to maintain the uterus lining, so it keeps it ready for an embryo to implant if you do become pregnant. So again, when you've got high levels of progesterone, it inhibits the production of FSH and LH from the pituitary gland. So you get that inhibition or the slowing down of production of FSH and LH so that the pituitary gland isn't producing those hormones and isn't maturing and releasing eggs. Okay, so here's a really nice summary for you to help you remember what the reproductive hormones do and where they're made. So you need to know the names of the reproductive hormones, FSH, LH, estrogen, progesterone, where it's produced, so either pituitary gland or ovary, and their main functions. If you're doing the higher tier, you also need to learn what their interactions are with each other and with all those other hormones. So if you've learned that slide, basically you'll be able to answer the majority of questions about reproductive hormones because you'll have a really good understanding. If you all are doing the higher tier, it's not been very long. It's been quite a while, in fact, since we had a graph which sometimes shows the concentration of hormones in the body. So it's worth actually making sure that you have some sense of an understanding of when all of these hormones are released. in the menstrual cycle. So, and they all interact with each other. And these interactions are all called part of a negative feedback loop. And a negative feedback basically means that you are reducing levels when they rise and increasing levels when they fall in order to maintain the optimum concentrations of these hormones. So if you see on this graph, you know that day 14 is when ovulation occurs. So you would expect a peak in the concentrations of FSH and LH. So remember, the FSH hormone matures an egg and LH causes it to be released. So the peak of LH is just slightly after FSH, and that release means that the egg is released in day 14. So you can look at those two peaks to work out which hormone is which on any graph. And if you think about building and maintaining the uterus lining, you'll see this level of estrogen rise up to build the lining, and then estrogen is no longer needed because the lining's been built up. So we need to start increasing the level of progesterone, which maintains that uterus lining, okay? So you can see that estrogen, progesterone, LH and FSH are constantly cycling and fluctuating throughout the menstrual cycle. We can also manipulate the menstrual cycle in order to treat people for fertility treatment. So if you're trying to have a baby, one way you can do it is by increasing the number of eggs you produce. So if you can give injections of FSH and LH, you should mature and release more eggs, and that increases the chances of getting pregnant. These two hormones are also really important in the first stage of in vitro fertilization. That is when fertilization doesn't happen in the oviduct, it happens in a dish in a laboratory. The procedure for IVF is really as follows. They give the hormones FSH and LH, given by injection to the woman, and that will stimulate several eggs to be matured and released. And these eggs are then collected along with some sperm from the male. The eggs are fertilized by the sperm in a dish in the laboratory, and that are often kept at room temperature, body temperature rather. And these fertilized eggs are then left in the dish at body temperature so that the eggs can divide by mitosis to form an embryo. When the embryo is around 8 cells in size, 1 or 2 of them may be transferred into the mother's uterus at the correct time in the menstrual cycle. So that's normally after day 14, where if a natural fertilization event would happen, it's around then. There are obviously lots of advantages to this fertility treatment, whether it's IVF or increased FSH and LH. It will allow couples to have a baby who might not conceive without the support from the extra hormones. Allows same-sex couples or women without partners to have babies. That sometimes does involve a surrogate, which is somebody who will carry the baby for you, but you could actually carry out IVF with a sperm donor if you don't have a partner, and you can put your eggs in with that sperm and then have that embryo inserted back into your own uterus. It also allows couples to freeze embryos so they can still have their own children. For example, if they're having chemotherapy or cancer treatment, it might be that they want to go through embryo formation and freezing and then try and have a baby after the treatment's over. Disadvantages, though, there are some ethical considerations thinking about any unused embryos. So if you use one of the embryos, if you make 8, for example, and you use one and have a baby, do you want to have 7 other children? So it's decisions about what to do with those embryos. Should they be used in stem cell research or destroyed? How do people consider that as a life or not? It's also emotionally and physically stressful. Being injected with those hormones can be quite emotional for the mother. The success rates are still quite low, so relatively not really made lots of improvement with success rates. And there's a risk of multiple births if you implant lots of embryos. So now they tend to just implant 1 or 2 embryos to reduce those risks to the mother and the baby of multiple birth. But sometimes you don't want to get pregnant, and that's where you need to use contraceptive hormones. So you can use those reproductive hormones to make contraceptives. The contraceptive pill contains estrogen, and that prevents the eggs from maturing because, remember, estrogen acts as an inhibitor of FSH. So you'll stop eggs being matured in the ovary, and if an egg is not matured, then there's less chance of it being released. But to prevent it being released, you could add progesterone to your contraceptive pill, and that will stop it from being released because progesterone acts to inhibit both FSH and LH. So an estrogen and progesterone combined pill would actually stop eggs maturing and being released. Now, when they first used the combined pill, the high doses of estrogen led to side effects like headaches and breast tenderness and mood swings and increased blood pressure and blood clotting, none of which sound very much fun. So they started to use low dose or no estrogen pills, so a progesterone only pill, and they were found to have fewer side effects and to still work effectively. If you're doing the higher tier, remember, you do have to know how that progesterone only pill works, because you need to know it inhibits FSH and LH, so no eggs are matured or released. And it also, the progesterone only pill will actually maintain the uterus lining. Often when you're pregnant, high levels of progesterone are present because you want that maintained uterus lining. You don't want that to start to be degraded and come out in menstruation. But you can get many different types of contraceptives now which do contain the hormone progesterone. It doesn't have to be the pill. It can be an oral pill, which you have to remember to take every day. So there is that you do need to remember, take it there every day at around the same time. To avoid that and increase the chance of remembering to have the progesterone, you can have a patch. They tend to be left on the body for a bit longer, but you do have to regularly change that. Or you can have an injection that tends to last about 3 months. So you can have 3 months worth of contraceptives, but you do have to go to a clinic to have that injection. You can have an implant, which is shown in your arm just there, a little plastic rod which contains progesterone, which is released slowly, and that can stay in, I think, for up to 5 years now. So, but you do have to have a small surgical procedure in order to put that in place. You also need to have a surgical procedure to have an IUD, which is called an intrauterine device, and this just sits inside the uterus, and that coil helps to prevent pregnancy, but it also releases slow release progesterone. So intrauterine devices can also help to prevent the implantation of any embryo. So it's a really effective contraceptive, as well as the progesterone that's also there. And there are lots of advantages of progesterone-only contraceptives. They often make menstrual bleeding lighter and less painful. It can reduce your risk of some cancers, can reduce your risk, symptoms of PMS, so premenstrual syndrome, and those effects of that, as well as acne. And it may reduce the risk of non-cancerous breast disease. The disadvantages are it can cause temporary side effects, especially at first, like headaches and nausea. It can increase your blood pressure. It doesn't protect you against sexually transmitted diseases, so barrier methods like condoms will prevent the STIs, whereas progesterone-only contraceptives just protect you from being pregnant. And there's been an increased link to an increased risk of serious health conditions like blood clots and breast cancer. But obviously with any drugs, as you learnt in B3, any drugs, there are always side effects. So it's some of these disadvantages are actually disadvantages from pregnancy. For example, you are at greater risk of getting a blood clot when you become pregnant. There are other non-hormonal methods of contraceptives. Obviously, you'll be aware of a lot of these from your personal development lessons. So you'll see condoms are very effective at preventing sperm reaching the egg. So that's a barrier method, and they're also very effective at reducing STIs and the transmission of diseases. Diaphragms are plastic cup-shaped devices which can block the cervix, so the sperm can't reach the egg. So again, is a type of barrier method. Spermicides are chemicals which you can apply in a gel which kills sperm. You can also try abstinence at certain times of the menstrual cycle. That means not having intercourse when an egg may be in the oviduct. The problem with that is not everybody's menstrual cycle is exactly every 14 days when an egg is released. So that is kind of a higher risk of pregnancy using that method. And you can get surgical male and female sterilization. So that's often called a vasectomy and a hysterectomy, which is the removal of the organs or closure of the organs that the sperm can pass through or the eggs pass through. Okay. So that's really it for B5. There's quite a lot of information in there, but it's certainly a topic that if you know a lot of this information, you will be able to apply it in the exam. So all that's left is for me to wish you good luck and hope your exam goes well. and have a look back through the video if you need any extra information. Look on some Quizlets or exam questions in order to develop your understanding a bit more, and hope it all goes well.